I'm Vijay Ramanan. I'm a behavioral neurologist and neuroscientist at the Mayo Clinic in Rochester, Minnesota. And I'm delighted to share some imaging genetics work relevant for Alzheimer's and related diseases. This is a team effort with Prashanthi Vamuri and other excellent colleagues, and the results are now published in Brain Communications. Over the last decade plus, our field has developed and validated non-invasive PET imaging methods to assess during life the burden of amyloid and tau in the brain. These are the two toxic proteins that build up in the brain in Alzheimer's disease. One door this has opened up from a research standpoint is to be able to do this biomarker imaging in large numbers of people, and then be able to ask the question, what inherited factors may predispose an individual to be either more susceptible or more resistant to building up those proteins in the brain. In particular, given that the burden and distribution of tau in the brain is very closely related to cognitive symptoms and impairment in Alzheimer's disease, that makes it a very ripe target to study, as we did in this work. This paper I'll briefly talk about describes what is, to our knowledge, the first published genome-wide association study of tau PET. The graphical abstract here gives an overview of our approach. We analyzed a population-based sample of older adults, all of whom had tau PET imaging and genome-wide SNP genotype data. The goal was to discover genetic variants called single nucleotide polymorphisms, or SNPs, which were associated with tau PET burden. Through this genome-wide association study, we discovered two variants, one on chromosome 5 and another on chromosome 7, which showed genome-wide significant association with tau PET burden. In both cases, the presence of the minor or less common allele at that gene position was associated with higher tau PET burden. And the pattern of that association was in a very Alzheimer's-like topography on the brain, as shown in these pictures. The brightly colored areas in the temporal, parietal, and posterior cingulate parts of the brain indicate regions where presence of those variant alleles had the strongest association with higher tau PET. The implicated genes in these associations have some interesting potential connections to AD. The hit on chromosome 5 was in a gene called PPP2R2B, which is highly expressed in the brain and regulates the activity of an enzyme family known to control the phosphorylation status of tau. The hit on chromosome 7 was in IGF2BP3, which has been studied in relation to several types of cancer, including glioblastoma, and which participates in insulin-like growth factor pathways, which have been proposed as neuroprotective in Alzheimer's. As we discuss in detail in the paper, there are important limitations of this work, including the frequency of the variants discovered and characteristics of the sample and the tau PET tracer used, among others. But we hope this work will facilitate and stimulate other similar studies in independent cohorts that will allow for opportunities to demonstrate replication of these findings, and more broadly, to advance knowledge about the factors that influence tau accumulation in aging and degenerative disease. Thanks very much for watching. Hope you enjoy the paper, which is now published in Brain Communications.